the receptor here, and the RAS, which is the first relay protein, and all these other relay proteins, and the S-phase enzymes. If any of those, the genes for any of those proteins mutate in such a way so that those proteins become constantly active, because remember, normally these proteins are in an inactive state until they get activated in the signal transduction pathway. But if the genes get a mutation so that the protein is stuck in a constantly active state, then the cell is going to be stuck in mitosis. And imagine this. Imagine that the receptor mutated, the gene for the receptor mutated so that the receptor protein is now constantly signaling the cell, even though there's no growth factor present. Well, the RAS protein is then going to say, okay, receptor is activated, so I should get activated. So they call these oncogenes when the proto-oncogene is mutated to be in an always active state. So anyway, if the receptor becomes an oncogene, then it's going to activate RAS, which is the first relay protein, and then RAS is going to activate the next relay protein, which activates the next relay protein, which activates the next relay protein, and away the cell goes, stuck in the cell cycle. But it doesn't just have to be the receptor. Imagine RAS itself mutated to become an oncogene. Well, here's a thought question. Would this stick the cell in mitosis? Would the cell get stuck in mitosis now? It would, because if RAS is constantly stuck on the active state, then it's always going to be activating the next relay protein, which is always going to activate the next relay protein, which activates the S-phase enzymes, and around the cell it goes again, stuck in mitosis. All right, if this relay protein were to mutate to an oncogene, does the cell become cancerous? Yeah, it does, because it's always going to activate the next relay protein, which activates the S-phase enzymes, and around we go. And so notice that that rule of thumb applies to any of these. If any of the genes in this mitosis pathway mutate to oncogenes, if they become always active, then the whole pathway is going to be constant. Well, I shouldn't say the whole pathway, but eventually the S-phase enzymes are going to be activated, and so the cell will become cancerous. So there's more than one type of mutation in these proto-oncogenes that can give you cancer. If any of the genes in the pathway mutate to an always active state, the cell can become cancerous. You buy it?
Susceptibility to mutation, does that vary between persons?
I'm sure it does. I guess I don't know the specifics of it, but all of our physical traits, any physical trait you can think of, is going to vary from one person to another. So I'm sure some people are more susceptible to cancer than others based on their genetics. Okay, so just to recap that, many cancers are caused by these proto-oncogenes mutating to oncogenes, which is basically an always active form of the proto-oncogene. Now, there's another type of mutation that can lead to cancer, but it's not a mutation for one of these oncogenes. It's a mutation in things called tumor suppressor genes. And to understand these, maybe I should make an analogy here. The proto-oncogenes are like the gas pedal system in your car. I don't know exactly how my car operates, but there's the gas pedal, and when I push that down, it must push a little rod somewhere which allows more gasoline to flow inside the engine. Somehow that leads to the car going faster. And so all those parts of my car that are the gas pedal or connected to it are basically there to make my car go faster. And so that's what proto-oncogenes are. They're part of the gas pedal system to make the cell divide more. The tumor suppressor genes are more like the brakes of the car. Their purpose is to slow down the car. So your cells have built into them proteins whose job it is to inhibit mitosis, to keep the cell from dividing. And so those are like the brakes. And so when those mutate to a loss of function, like losing a function of your brakes, then that would tend to make your car go out of control. In other words, you can get cancer if you lose tumor suppressor genes. So let's look at those guys a little bit. Tumor suppressor genes are genes whose normal function is to prevent mitosis, to inhibit mitosis from, so it doesn't occur in an uncontrolled manner. And many of these tumor suppressor genes are inhibitors of the proto-oncogenes. So let's look at this. Here's this pathway again, and let's watch it operate. Here comes a growth factor. Growth factor activates the first relay protein, which is normally this RAS protein, which activates the next relay protein. Now, remember, activation of this relay protein is supposed to end up activating the next relay protein right here. And up to this point, I've been showing you where each relay protein typically kinases the next relay protein to activate it. But in some cases, it operates slightly differently. And this is an example of that. That protein, there's the next relay protein in the inactive state. This protein right here is an inhibiting protein. In other words, this relay protein's natural state is to be active, but it's kept in an inactive state because this inhibiting protein is binding to it and stopping it from being active. And so that previous relay protein that I was showing you activates the next relay protein by removing the inhibiting protein. Just kind of pulls it off. And now that next relay protein is no longer inhibited, and so it becomes active and can go off to activate the next relay protein. So, I think I have to go, there's a bit of text that I skipped a minute ago. Here we go. When I was talking about cells getting back into the cell cycle from GO, I said most relay proteins activate the next one because they're kinases. Some relay proteins activate the next relay protein by removing an inhibiting protein that was inhibiting the next relay protein. Well, what I'm saying here is those are tumor suppressor genes. Many tumor suppressor genes are those inhibiting proteins that keep a proto-oncogene in an inactive state. And so when the growth factor has started this pathway, the growth factor leads to the temporary removal of that tumor suppressor gene from this this proto-oncogene here. And so that allowed this proto-oncogene to be active for a little while to activate the S-phase enzymes and then put the cell in mitosis like it's supposed to. But remember, mitosis is always done in a controlled manner, and so as soon as a round of mitosis has occurred, the tumor suppressor gene is put back on to that particular proto-oncogene, and that inhibits it again, and that's what turns on. That's what keeps the mitosis from being stuck in a always mitosis mode. And so as long as this tumor suppressor gene is there, the cell is not going to be stuck in mitosis. The cell is going to be able to stop itself. But the problem is this: if a mutation occurs that stops the tumor suppressor gene from being active, in other words, the mutation inactivates the tumor suppressor gene. If we get rid of this tumor suppressor gene, then there's no more brakes on the system. And that proto-oncogene that it's supposed to be inhibiting is no longer going to be inhibited. And so the cell is going to be stuck in mitosis. So just to recap these, there's actually two types of mutations that will give you cancer. One is where the proto-oncogenes get mutated so that they are constantly active, kind of a gain of activity mutation in the proto-oncogene, because those are like the gas pedals in your car. And if something happens to your gas pedal so it's always stuck in the too active position in your car, it's going to go, you'll be out of control. But also your car can go out of control if you lose function from the brakes. So gain of function for proto-oncogenes gives you cancer, but loss of function, inactivation mutations in tumor suppressor genes can also give you cancer. Sound goody? All right, now there's a few other proteins that are considered tumor suppressor genes, but that they are not inhibitors of proto-oncogenes. So what are these tumor suppressor genes that are not inhibitors of proto-oncogenes? Well, remember the definition of a tumor suppressor gene is a gene that in some way normally prevents mitosis from occurring in an uncontrolled manner. So any gene that when it loses its function, you tend to get cancer is a tumor suppressor gene. So let's look at some of those other types of tumor suppressor genes. Some of these other types of tumor suppressor genes are DNA repair enzymes. Let's say this is part of your DNA, and let's say that a mutation has occurred for whatever reason. One of your bases has changed like this. Well, the body wants to repair that because it's a mutation in the gene, and there are DNA repair enzymes. These enzymes constantly scan along the DNA looking for mutations that don't match up with the opposite strand, and when they do find them, they try to repair them. Sometimes the DNA repair enzyme might not know whether the G or the T was the wrong one, but in some cases it can essentially guess and correctly repair the mutation. So that's fine. Now, let's say that another mutation has occurred. Yeah, there it is, a T right there. All right, so here comes the DNA repair enzyme. But the problem is the DNA repair enzyme is itself made from a gene somewhere in the genome. And if the gene for this DNA repair enzyme gets mutated in such a way that we lose the DNA repair enzyme, the DNA repair enzyme's not made anymore, well, then there's going to be nothing inside that cell to correct the mutation. The cell's not going to be able to repair its own mutations anymore. And matter of fact, it wouldn't just be that mutation. Mutations happen all the time. And so the cell would start to collect many different mutations. The DNA would get more and more mutated over time if there's no DNA repair enzyme. And I think what that means, well, if the cell is experiencing more and more mutations, then it's more and more likely that you're going to get a mutation that gains function in any of these proto-oncogenes, like here and here and here and here and here and here. And it's also more likely to get a mutation that inactivates the tumor suppressor genes, and both of those can lead to cancer. Activating mutations of proto-oncogenes and inactivating mutations of tumor suppressor genes. And so it just comes down to the fact that anything that stops the ability of the cell to repair its DNA, to repair mutations, is going to promote cancer because you're going to get more mutations in proto-oncogenes and tumor suppressor genes. All right, so let's see what's going on here. There's yet another type of tumor suppressor gene, but it's not a DNA repair enzyme. Let's say that the cell is going through the cell cycle, and somewhere in the cell cycle, let's say right here, some of the DNA gets damaged. Maybe you were out in the sun, you got a bad sunburn, and the ultraviolet light damaged some of your DNA. There's a protein which is considered one of the tumor suppressor genes called P53, protein 53, that blocks the cell cycle from passing the G2 checkpoint. In other words, this P53 protein is essentially the working part of the G2 checkpoint. Remember, this checkpoint is about halting the cell cycle if the DNA has been damaged. And so P53 does not actually repair the damage itself, but P53 will stop the cell cycle until the damage is repaired. It gives the DNA repair enzymes time to repair the damaged DNA. And that way the mutations don't get passed on into the daughter cells if the cell were allowed to go past the checkpoint there. P53 is sometimes called the guardian of the genome because that's what it does. It halts the cell cycle until the DNA can be repaired. Well, the problem here is P53, the gene for P53 itself can be mutated. And so if the cell loses the gene for P53 through mutation, then the cell's going to have more mutations. Again, not because p53 actually repaired mutations, but p53 gave time for the DNA repair enzymes to function by halting the cell cycle there at the G2 checkpoint. And so without sufficient time to repair the DNA repair enzymes can't fix it, and well, you get more and more mutations. And so loss of p53, you end up with more mutations accumulating in the cell's genome. And then, like I said before, if the cell is collecting more and more mutations, it's more and more likely you're going to have a mutated proto-oncogene or tumor suppressor gene, and that's going to increase the likelihood of cancer. So since loss of p53 ends up giving you more likely to get cancer, p53 is also categorized as a tumor suppressor gene. Alrighty. All right, some final closing thoughts here about cancer and the molecular biology of cancer. So notice that one of the major points of this was that cancer is all about mutations. Mutations in the proto-oncogenes that make them too active, or mutations in tumor suppressor gene that take away their activity. And anything that causes mutations is called a mutagen. There are certain chemicals